same position on that gene. I know what I'm doing here, okay? So that's why we use DNA. Now, the thing that's interesting about plants in particular is that unlike animals like us, we have two sources of DNA, your nuclear genome and your mitochondrion. In a plant, you actually have three sources of DNA, the chloroplast, the mitochondrion, and the nucleus. Here's their relative sizes, kind of small compared to the nuclear genome. These organelles have small amounts of DNA, but for a long time, this was the workhorse in plants. There were a lot of cool features about the DNA in chloroplast that made it really nice to use. I'm not going to bore you with all the details, but you can sequence the entire chloroplast genome with no trouble. Okay, if you wanted to do this, we could all get together, and within a week we could sequence the entire chloroplast genome for a bunch of plants. Okay, and here it is. And you don't need to remember the structure. I don't really care, but that's what the chloroplast genome used, and it was used a lot because it was just really, the structure was so conserved. All the genes were just in one copy. We knew a lot about it, useful at a variety of levels from the species and deeper in time. But the mitochondrial genome was not so good. It was not so useful because it was pretty complex. So my point is, a lot of the phylogenies you're going to see in your textbook, you will not think about this very much, but I'm just telling you, they were based on sequences of chloroplasts or genes in the chloroplast. Probably now the entire chloroplast because it's so easy to sequence. I mean, you guys have all heard of 23andMe, right? You can sequence your entire genome for less than $100. It's amazing. The first, how much, does anybody know how much the first human genome cost to sequence? Millions of dollars. Some people thought it was a waste of time and money. And now think of how a fundamental part of your life it is. Just a fundamental diagnostic tool will be your complete nuclear genome sequence. But we started out in the plant world just by sequencing chloroplast genes. In the animal world, they sequence mainly mitochondrial genes. Now, with more and more technology, we're doing more and more with the nuclear genome, but it's just harder to use. The mitochondrial genome has a lot of bad features, so we don't always use it. There's incredible variation in its size, so it's not as often used. So, let's see, how are we going to finish? All right, so as I said, we keep making advances in DNA sequencing. There was next generation sequencing and then next generation sequencing by 2010. And we just keep getting these amazing toys, which you can roll out and have one in home for probably just like $500,000. There's nothing to it. Like, this would look really nice next to your refrigerator, right? These are actually old. This is an old slide, but I kept them in because they have such cool names. I'm going to take my Ion Torrent for a test drive this afternoon. This is already out of date. The 454 is out of date. These are ones we still use a lot that would be considered next generation. Illumina and PacBio, we have these on campus, and really this and this, the Illumina would fit on this desk, okay? So what my point is that something that powerful that can generate so much data is, the size of a cabinet. It's amazing. So the era that you're living in is it's mind-boggling. And I guess I always think of this line from one of the old Batman movies. This is one of the old Batman movies, and I think it's such a great line. He looks up, this is the Joker, of course, right? In that case, it's Nicholson, and he looks up at Batman who's going over in the is that the bat plane? Yeah, and he says, where does he get such wonderful toys? And I think that is where we are in modern science and where we are in systematics. You have at your disposal so many incredible toys, like these guys, that we can use to generate data. And that is all part of the revolution that has taken place in the field of systematics. Okay. So here's, just to give you an idea, here's how much data you can generate in a day with like the HiSeq. Let's see. I don't have, oh, here we go, single reads. You can get 3 billion with the HiSeq, and this is how long they are, 100 base pairs. So, you know, the human genome is 3 billion base pairs. So, you know, basically there's no trouble generating your entire genome in a day with one of these machines. That's why it's 99 bucks. Okay, so far, so fast, these technological advances. Here's the price tag, by the way, of the first human genome. And it took over a decade to sequence and assemble, and it wasn't very good, the first publication. It was kind of a crappy assembly. $2.7 billion to do that in 13 years to produce it. Now you can do it with $99 for 23, maybe less than that. Has it gone down? That's the last time I checked, it was under 100 bucks. So the impact on this sequencing has similarly been so huge and systematic. So we routinely sequence the entire chloroplast genome. We routinely sequence now many nuclear genes, and now even entire nuclear genomes in plants, not just, you know, numbers and numbers, hundreds and hundreds of plants. So that has made this really a reality. With just a few billion more base pairs, we can nail down this phylogeny. The whole thing is accelerated over 20 years from just a few, we can do a few genes to now, we can just do the entire genome. In fact, there is a new project that is coming out of China that they are funding, and it is called the 10KP project, meaning 10 kp, meaning 10,000, and they are going to sequence 10,000 plant genomes, the nuclear genome, the entire nuclear genome for 10,000 plants. Inconceivable not that many years ago, all right? But now this is a project that will take place over the next 5 years. So my job here is just to try to hopefully kind of blow you away with what is going on today in the field of systematics. I mean, I've been at this a long time. It's by far, I always say this, I mean, this is the most exciting time ever, but this is really like the most exciting time ever. So your textbook is trying to keep up and reflect that. Those phylogenies that you see and that we will look at as we go through group by group by group are largely based now on, well, they are based on DNA, and they're changing. So some of the times we're going to go over groups and what's in the textbook. I was like, you know, there's just a phylogeny that came out last week, and it was based now on, you know, way more sequence data, and we have a different view of these relationships. So we'll give you sort of the up-to-the-minute view of how things are related. Are there questions? Went over a lot of stuff. Okay. What are the main things you want to remember? Some new terms today. Porology. What's porology? What's a porologous group? Remember, walk out knowing this. Oh, I'm sorry, paraphyletic. Yeah, paraphyletic, sorry. I'm thinking of genes, yeah, thanks. That's another term, we won't introduce that until, maybe we'll just stay away from porology altogether. Paraphyletic group, yeah, are those good things? No, we want an ancestor in all of its descendants, and that is, what's the correct term for that? A monophyletic group or a clade. Okay, you can use either one of those. Homoplasy, what about it? Similar features, they could be derived in parallel, right, independently, or reversible, right? Through time, change, but they revert back, a lineage reverts back to the ancestral state. Those are things that happen in the course of evolution. The cool thing is now with all these DNA data, we have a better ability to figure out where those occurred with morphological features. All right, why is DNA so good? It's cool, that's one. Lots of characters. Homology. It's really easy to figure out, okay, yeah, that goes with that. That's a gene, okay, I can compare those. It's harder to compare things like hairs, leaves, pollen, much more difficult to compare. And in the animal world, the same thing. You know, how do you compare some of those structures that you're looking at? Very difficult. Okay, anything else to add? We're good. Oh, and anybody who came in late, there was a problem. I guess that Tree of Life website crashed over the weekend. So if you were unable to do that, that's the reason. It worked up to about Saturday, we think, and then so we'll talk about this again on Thursday. But right now, that means it puts off, unless it comes back up quickly, it puts off your homework assignment. Okay, everybody good?
I don't want to
Okay.
walk around, so I got my scooter out.
I'm sorry, it's too far apart.
Oh, I forgot to say, somebody asked about textbooks.